Bismillahirrahmanirrahim. I was talking the muscular system as far as general anatomy part of it, and I'm continuing these lectures further, so you can understand clearly from A to Z about the muscular system from all aspects. So I have reached actually up to this slide. That this is the slide which I have shown previously. Starting with the external coverings and all the aspects has been discussed, and then microscopic anatomy I have discussed, and then we have the structures inside the muscle. The functional unit is called the sarcomere, and the constituent of the sarcomere was described. In the morphology is concerned, in the what is called as the light microscope and the electron microscope. So this is what I have given you the details of it. The actin and myosin. Myosin at one end there are two heads. It grips with the actin and it slips down like this. So they are crossing over or slipping each other will cause contraction of the muscle. This was the concept. Inside, there are proteins of the myosin and the protein of the actin, which are the organic material inside. And it will be seen in the detail of the microscopic anatomy. The physiology must have taken out that the skeletal must be stimulated by a nerve, motor neuron to contact. Let's start from here. Downgoing, the signals comes here. And through the transmission of the nerve impulse to the muscle. The first step is the nerve, which is depolizes now after depolization transmitter, neurotransmitter. It is one of this is called acetylcholine, which is present in the skeletal muscles only, but not in the smooth muscle. There is another a way to stimulate the muscles of the smooth muscles or cardiac muscles. Acetylcholine is the only neurotransmitter which is yet detected at the neuromuscular junction. It is a synaptic, and the synapses occur here. You see the terminal, axonal terminal motor unit, synaptic vesicle containing the acetylcholine. It is releasing it after deposition, and then it will act on this on the muscle myofibril, and then it will contact as a part of the myofibril, you can say. This is shown here. And then actually the second step in which I stopped there, because now the concept it was a very old one. The concept is a very old one, although that now this is which is the step 2. I stopped there. It is old one that at one time they said only receptors are there, then the details have been given by the anatomist and the histologist, and afterwards the organic chemistry people who are definitely working as a researcher. They said that afterward that only the receptors is okay, but what about the specific receptors are concerned? There are different receptors. You know that about it. I will take another class with the receptor is concerned. Now they go deeply inside. And see, there are actually the concept has been changed now. And they said there is inside the cell, for the entry is concerned, anything which is entering into the cell will definitely, they said that they should go to a proper channel inside. Otherwise, that cations or anions, whatever it is, because there is exchange of the ions there. To contract the muscle, definitely the calcium is needed here. So, these muscles, neurotransmitter causes the muscle membrane gates now to open, and the influx and efflux of the anions or cations. Now, this is the problem here. What is the gates to open? What are your gates? It is should be stopped here. That the gates is related with the ionic channel. Every cell has got a definitely channels, and channels they are specific for specific cation or a specific anion, and that is called the ionic channels. Actually, it was written in the physiology, but to me at one time when I was a student or even in the FCPS examination, this concept was not at that time emerged. Remember, it is new for me. What is the ionic channels and specific channels, how they work, etc. So I stopped there because I have to stop this one because it should be cleared from A to Z. Problem is this. That's seen in step 3 that the sodium-potassium exchange takes place, causing the sarcolemma reticulum. Then it releases the sarcolemma reticulum to release the calcium ions. It is step 3. Step 4, this release of the calcium ion starts the muscle contraction as the action filaments slide past the myosin filaments. Already mentioned out. Now this crossover bridging or shortening, and the myosin has got two heads. It grips the actin and slides on each other. It occurs because of the calcium release. And then there was one of the myofibril will contract in this fashion. A bundle or more than that, fascicle, the whole muscle will be contracted like that accordingly. It is the voluntary action. Muscles are skeletal muscles are involuntary. And you have to do voluntarily these skeletal muscles to stimulate. Problem here is this one. What is the gates? Obviously, this is written, but it is not clear here. So, effort has been made by me to clear this by this. That is the. Let's start some discussion. Although this lecture is quite long, it will take time. No problem. I am Professor Ali Azhar. I am speaking as a facilitator of this tutorial, as a global honorary professor of medicine without name or without fame or without any economics. It doesn't relate with me. So this is new for me or any person who has passed his examination, FCPS, I have passed it in 2003, 4. It is 2000. No, it is 2002, 2002. So afterwards, after 210 to 2011 or 2010, now this concept has become definitely emerging. Ionic channels. In 2004 or 3, actually, they have described first time there is a channel by a chemist, and who obtained Nobel Prize for it in the chemistry. Because chemistry and the medicine need chemistry to understand. Remember, you cannot say that I have actually read Raad Gai, Baad Gai, organic chemistry and inorganic chemistry and other things, or related with the physics, their biophysics, etc. You have already forgotten it. Problem is this. So this I have to take this to clear this concept which I am taking. Now let's discuss the ion channels. What are these ion channels? The ion channels or ionic channels. Now they are pore-forming membrane proteins. They actually channels are pore-forming membrane proteins that allow the ions to pass through the channel pore. Their function include establishing a resting membrane potential afterwards, shaping the action potential and other electrical signals by gating. The gate is opened and then closed. The flow of the ions across the cell membrane. Now, this is the concept of the gate. It means that it's shut down after the action because you do not need it now. Controlling the flow of the ion across the secretory and epithelial cells, they are functioning there also, and regulating cell volume. Ion channels are present in the membranes of all cells. Remember, membranes of all cells. Ion channels are one of the two classes of the like a protein that they are dependent on ionic exchange proteins. The other being the ion transporters. They are the proteins themselves working. And one another type of the ion are transporters. They are basically going just like the active transport, for example. You need ion. The study of the ion channel often involves biophysics, electrophysiology, and pharmacology. While using technique including these are the techniques that they have actually emerged during the last 20 years. The voltage clamp, patch clamp, immunohistochemistry, X-ray crystallography, fluoroscopy, and RT-PCR, the reverse transcriptase, polymerase chain reaction. Their classification as molecule is referred to as far as molecular concerned, they are called genomics, channel omics. Now this is the subject actually which was introduced. Now these subject is in the process of development, and a lot of people are doing their PhDs in this, the channelnomics, because this is a very important concept for drug action or anything you want. Channels are there. They have got gates. Gates are open and close. The ion influx or efflux, gate open and close, and forming they form first of all the membrane, resting potential, and then action potential. Afterward, because if this gate is it is closed, now the action should be started out according to the definition of the action potential. Now, the schematic diagram of this is, this is what is seen here, and the person got this one. The channel domains means channel 1 channel domains. Domains are the molecules which are passing, or a group of molecules, typically 4 per channel. And one channel can afford 4, not more than that. This is here. It's a cross section. It is longitudinal section of any channel is concerned. This is the cell membrane here, and this is embedded here. This is the channel, which is a concept of it. And then we have got the vestibule, outer vestibule. This is the outer vestibule, this one and this one. Selectivity filter, it is there. This is the selectivity filter with a region of this of the vestibule, and this way this gate opens or closes. Selectively filter. These gates are opened, filtered it, and going down or outside, inside or outside. So sodium-potassium exchange depends on this channel, which is the ultimate important part for the efflux of the calcium afterwards. Again, the calcium has got a specific channel. In this fashion, as sodium as potassium is concerned, they are also dependent on the channel, the specificity. And then fifth channel is phosphorylation side. This is the phosphorylation side. It is filtered out. It should be phosphorylated accordingly here, and then it stimulates it is stimulating the cell membrane itself, and inside the what is called the ectoplasm is concerned. In this fashion, the deposition occurs, and the action is dependent on the sodium-potassium exchange, filtered out, phosphorylation occurs, and then it acts. The another channel will give the efflux to the calcium ions, the outside. In this fashion, the action potential degenerated, and the action will take place. This is purely a very, very good example of advanced, physiology, or what is it called as the histopathology is concerned. Cytologists, they have done a lot of work, very hard work. You can't imagine it, how they are working. This is the main concept actually. The thing is this, the people do not know this. There is your domains, and then the outer vestibule, selective filter, diameter, selectivity filter, and phosphorylation. Then cell membrane will definitely depolarize it after the outflow. The resting membrane potential occurs, If a flux of calcium occurs, then active potential will develop, or action potential will develop. So I repeated it down 2 times, that's why. If you can understand me, what I am saying. Basic concepts is that there are 2 distinct features of the ion channels that can become differentiated from other types of the ion transporter proteins. The rate of the ion transport through the channel is very high. It is often 10⁶ ions per second or greater. It is much higher, trillion ions. Other than the other transport proteins are not like this. Their content is lower down, not higher content. They can't do the transporter type of a function in this type of influx. Then ion pass through the channel down the electrochemical gradient with a function of the ion concentration and membrane potential, downhill without the input or help of the metabolic energy. They do not need it. ATP co-transport mechanism or active transport, etc. mechanisms. They do not need it as far as any energy is concerned. No. Ion channels are located within the membrane of the excitable cells and of many intracellular organelles. They are often described as narrow water-filled tunnels that allow only ions of certain size and are exchanged to pass through. This characteristic is called the selective permeability. Specifically they are allowing, otherwise they do not allow. The gate is closed. No action potential, no generate, not at all. Because they are selective for the sodium, for the selective for phala, etc. The archetypal channel pore is just 1 or 2 atoms wide at its narrowest point and is selective for a specific species of ions such as sodium, potassium, as I have already said. However, some channels may be permeable to the passage of more than one type of ion, sharing a common charge, that is positive, cations or negative. 2 may be possible. Sodium, potassium, for example, often move through the segments of the channel pore in a single file nearly as quickly as the ions move through the free solutions. In many ion channels, passage through the port is governed by gate, and that is the gate I have already mentioned out. It's closed or open, which may be opened or closed in response to the chemical, electrical gradient, electrical signals. It depends upon the temperature. It depends on the mechanical force. And they are working on this, how the temperature opens, how the mechanical forces, etc., etc. They open the gate, and then the action takes place. This is the concept. Now, this is the 3-dimension picture shown here of the. This is the cell membrane. It is in, just like this, and this type of domains. They are setting like this. They are entering inside. All things are labeled here, and in this fashion you can understand that this is the part where the ions release. It is the part which is entered and then it is released. It is filtered out, phosphorylation. Then that action, resting action potential. If the flux occurs of the cations, then of course there will be action. This is the concept which is given in this very hard working they have done. Hard work has been done to organize this type of architectural concept. This is rather. The ion channels are integral membrane proteins typically formed as assemblies of several individual proteins, such as multi-subunit assemblies, usually involve a circular arrangement of the identical or homologous proteins. Assemblies of individual proteins, integral membrane proteins, and individual proteins, several individual proteins, assemblies, closely packed around a water-filled pore through the plane of the membrane or lipid bilayer. For the most voltage-gated ion channels, the pores forming subunits. Now they have also detected subunits, alpha subunit, while the auxiliary subunits are denoted beta, gamma, and so on and so forth. So there are a lot of these subunits are there in one channel to describe it. The biological role now. Because the channel underlies the nerve impulse, and because the transmitter activated channels mediated conduction across the synapses, as you've seen, the synapses, there's like a neuromuscular junction, or other than that, there are a lot of synapses are there. So the channels are especially prominent component of the nervous system because this is the highly excitable tissue. Indeed, numerous toxins that organisms have evolved for shutting down the nervous system of predators and prey, from the venoms produced by the spiders, scorpions, snakes, fish, bees, sea snails, and others, work by modulating an ion channel conductions or kinetics. That is why the poisons of different type of abomination, snakes, etc., they are definitely harmful to the body because they will modulate it. In addition, the ion channels are key components in a wide variety of the biological processes that involve rapid changes in the cells such as cardiac, skeletal, smooth, all excitable tissues, epithelial transport of the nutrients, and ions T cell activation in pancreatic beta cells. Insulin release is dependent also in search for new drugs. Ion channels are frequent target. New drug, remember. It is a new drug which will be. So, means that the diversity is concerned, 300 types of the ion channels are there, and they said that they are specifically gating are there, and depending upon the type of the gate, they open it, or they allow to the other ions to pass through. And there other things are there, localization of proteins. They should be specifically for this. And further heterogeneity of channel rises when the channel with different constitute subunits give rise to specific kind of current. There are subunits inside this channel, okay, which called subunit also, and they give rise to specific type of current that is we are feeling it, working, in a different, different part of the body or differently working, no problem. Anything which is there in the epithelial surfaces are concerned and so on. They depend upon the specific kind of current which is raised by subunits of channel. So the classification of the gating, there are voltage gate type, depending on the voltage gate sodium channels, for example, and the voltage gate calcium channels. And there are cation channels with sperm specifically, and so on. They are voltage gated, right? So, in this fashion, the channels voltage gated, the potassium channels and transport receptor potential channels, so on. It's a big discussion. I don't want to go in much detail of it, but there is conception should be there. And there are other gates are with hyperpolarization activated cyclic nucleotide gated channels, and there are voltage gated proton channels, and they are important for as the function is concerned of phagocyte like eosinophils or neutrophils and macrophages. So this is the concept which is given, and there are ligand gated. There are different type of channels which are ligand gated. Now, what is ligand? Ligand are the collection of molecules which is there specifically, and they can be recognized, which can be recognized by the receptor and then by the channels is concerned. Ligand gated are neurotransmitters basically, and it's whole metabolism is described here. I don't want to go in detail, but they are there. Specifically, the neurotransmitter, this is often located in the nervous system mainly, and they are lipid gated also. Channels which are called lipid gated. Phosphorin, protein gated and lipid gated and so on. So it depends upon lipid molecules which are there, in the plasma lemma. It is proteins with the lipid lipoproteins are there. So specifically, leak potassium channels gated by lipids, including the invert rectifier potassium channels. Now this is a channel which is invented or detected by the scientist. Salute to them, salute to them. And KC and Q potassium channel family is described. And this is a very detailed, as far as organic chemistry is concerned. Those who have have done their MPhil and PhDs in organic chemistry, they can understand, the details of it. But these information which are given by the chemists are very, very important to us as a physician is concerned and as a doctor is concerned. If you can understand, I don't know. These are my poor effort which I am going through. Other gating channels are also described by, for example, potassium channels, like invert rectifier potassium channels are there, and others are also there. Calcium activated potassium channels, and the tandem pore domain potassium, they are all described actually in the history. So in 2003 to up to 2023, about it is 20 years, and this is the study accepted afterwards, by the channels are concerned. Specific channels, how gated, non-gated, etc. The two-pore channels, lipid-gated channels, and the mechanosensitive ion channels, cyclic nucleotide-gated channels, etc. And temperature-gated channel also. This is an interesting situation in which the temperature is you are discussing a temperature. The temperature, if you are discussing, it means that thermoregulation and disease associated with the heat, etc. So, we might say that these are the specific channels, and depending upon the gates, specific channels, they are related with the gates, and there are certain channels which are not associated with the gates. Gate open and close. So only remember this. And there is, specifically speaking, there are specific for the specific proteins and cations and anions. Now this is the main thing you should understand. Otherwise they are working now the temperature gated, which are dependent upon the temperature. For example, receptors are present. You know that there are thermoreceptors which are, for example, present in the thalamus. Now, they are activated by this temperature gated channel. They are there. They should be there, the gated channels, because they recognize the signal, and then the negative feedback mechanism occurs, and the regulation of temperatures occurs, etc. That is the anti-rise centers are there, anti-fall centers. They don't know कि वो काम कैसे करते हैं. They don't know, and now they can understand. They are the temperature gated channels, specifically in the parts of the brain. And type of the ions, we are classifying also the chloride, specifically the potassium, and many type of channels, sodium alcohol channels, calcium channels, proton channels. And third type of classification is the cellular localization. You know that the function of the endoplasmic reticulum, the Golgi apparatus, is mitochondria. These are the compartments which are seen inside the cell. Now, it is also dependent upon these channels. And accordingly, they are plasma membrane channels and intracellular channels, etc. The Golgi apparatus is the major, thing where the proteins are assembled and finalized by the endoplasmic reticulum. And mitochondria develop the energy. These are the powerhouses. So they all dependent upon this. Inside, they are intracellular channels, or for the organelles. The organelle has got also membrane bound. If they are membrane bound, it means that they have described these channels. So what a hard work they have. The other classifications they have also done, and these are the transient receptor potential channels, which are seen in the Drosophila. This is a fly, actually specific fly in the countries where the phototransduction occurs in this. Depend upon this. The transient receptor potential channels, TRP channels, remain constitutively open, while others are gated by voltage. Other gated by voltage, but there are some channels which are not gate. They have no gate. They are continuously opened. Intracellular calcium influences pH, influences redox and osmolarity and mechanical stress, etc. These are the important points regarding their dependency is concerned. Detailed structure has also been described, and it is the Rodak Mechanicon using the X-ray crystallography. Now, this person has actually, probably he's Australian, I don't know, but it seems to be Australian, and he got the Nobel Prize in Chemistry in 2003. He's a chemist basically, organic chemist, and he described this channels in detailed structure. But other, about Berkeley Hill and Clay Armstrong, they also they have worked on these ionic channels hypothesis, and they proved it by crystallography. So this is the concept which is given. Now, in the last context and the last way to do it is the application of pharmacology. Now look how they are applied. Ion blocking channels, ion channel blockers. Ion channel blockers of organic and organic molecules. Like for example, the tetrodotoxin is a pufferfish and the nest of the defense. There are type of fishes are there inside. They have this type of toxins which is generated and killing the other fish by using the blocking of the channel. A sexy toxin, for example, dinoflagellate is a small parasite, and it will cause harmful to the others by blocking the ion channel. Some are voltage gated sodium channels, some are potassium channels, etc. These are examples. The application. The chondrotoxin, cone snails, lidocaine, UU novocaine, yes, they are now classified under this ion channel blockers. Local anesthetic blocks sodium ion channels. Remember, they are blocker of sodium ion channels. This is how they are describing it. And the dendrotoxin produced by the mamba snake is a very, very deadly snake. Mamba. The mamba snakes are dreadfully deadly. It's called a powerful neurotoxin, which can kill in 15 to 20 minutes. Mamba, which are usually seen in central and the South African zones. It is blocking the potassium channel. If you block the potassium channels, you cannot excite the tissue. It is not possible. And abidotoxin and other, scorpion, which is present in the eastern Indian areas, like for example in Madras and so on. The scorpion is present, it blocks potassium channels, and there is definitely paralyzing effect is there. And heteropodotoxin, which is the toxin which is seen in the heteropoda. Huntsman spider is a spider, which is a poisonous spider, and it blocks the potassium channel. These are the applications. Activators are also there. Activate like calcium channel openers like BAVE. This is a molecular name of it. Chloride channel openers like the phenanthroline, specifically. Potassium channel openers are the minoxidil. Now this is the minoxidil is used at one time as in cases of hypertensive emergencies. Minoxidil. It is a blocker which is called the alpha receptor blocker, very powerful. Now they are only using locally because the side effect is the hypertrichosis, and that is why using in cases of the baldness. The baldness is locally minoxidil solution is used. And sodium channel openers like DTT, dichlorodiphenoid, trichloroethane. This was a substance which is used in the agriculture department at one time. Now they are derivatives of the DDT. If the DDT is engulfed by the person, he will die because the sodium channel opens and it's open for many minutes, so much so that the hyperpolarization occurs and the patient will die because excitable tissue will not excite. Now in the disease is concerned, application in the disease. Like for example, these are diseases are known as the channelopathies. So this is the terminology we used, channelopathies. Now, equine hyperkalemic periodic paralysis, as well as human hyperkalemic periodic paralysis, they are caused by the defect in the voltage-dependent sodium channels. Another one is the paramyotonia congenita. This is a disease which is related in neurology, actually. Congenital disease, although, but it's a paramyotonia, the myotonic type of contraction is persistent and potassium aggravated myotonias. And generalized epilepsy with the febrile seizures plus means that these are the you have also learned in the epilepsy there is a generalized epilepsy in the as well as febrile seizures are concerned. This is related with the febrile. It becomes permanent as the age grows. They said you can apply channelopathy here. The specific channels are activated here. GEFS plus लगाया है इसमें तो it down the voltage gated ion channels and is slowing down the repolarization of cells. These all causing this phenomena. Repolarization is affected. Episodic attack is also example. Incoordination, bouts of severe incoordination with or without myoclonus is concerned. Means twitching, localized twitching is myoclonus of the face. Muscles are called myoclonus and can be provoked by stress, throttle, or heavy exertion such as exercise. This episode occurs because of the channelopathy. Familial hemorrhagic type of migraine is what is called migraine equivalent is a type. Spinocerebellar ataxia type 13, long QT syndromes, this is favorite subject of mine, and ventricular dysrhythmia syndromes, etc. They are potassium channels, which is affected in there, and all of which affect cardiac repolarization. So repolarization is defective. Brugada syndrome, again, the voltage-gated sodium channels actually are at stake, and gene mutations are there, and because it is a disease related with the gene mutations. Actually, it is the disorder of the voltage-gated sodium channel in the myocardium. Now, polymicrogyria, development in brain malformation, again it is seen in the just after birth, voltage-gated sodium channels are affected. Cystic fibrosis is very famous, it is chloride channel which is affected. Mucolipidosis type 4, it is the congenital, disease, mucopolysaccharidosis. And mutations which is seen in tumors, like for example the glioblastoma multiforme, it is grade 3 or grade 4 type of osteocytoma. Upregulation of the potassium channels and chloride channels enable the glioblastoma cell to migrate inside the brain. It will lead to the diffuse growth pattern, and the brain will destroy it. It's compressed very badly. And these are dependent upon the ion channels. The birth of an idea, I have already shown you in my, which was a moving lamp. Julius was commissioned by the Roderick Macon based on the molecule atomic coordinations. This is the channel, see this is the channel, this is the membrane, around it this is the membrane, and this is description of this, which I have already mentioned. What is Lambert-Eaton syndrome? Apply your knowledge of this now of ion channelopathy. Ion channel. It is a channelopathy or not? The Lambert-Eaton syndrome is, what is this? It is the result of antibodies against the presynaptic voltage-gated calcium channels. There are antibodies which actually block presynaptic voltage-gated calcium channels, and likely other the nerve terminal proteins in the neuromuscular junction. In this way, the connection between the nerve and the muscle that they supply. So the nerve where the depolarization occurs cannot be done properly, and the scene is that it is very, very similar to myasthenia gravis. But in this condition, if you will go, for example, 5 pound force to increasing to 7 pound force, is applied, so the power will increase rather than decrease. But in case of the myasthenic gravis, it will decrease. It will in the due time, 3 minutes, 4 minutes, the patient become tired. Here he will become more active. In the beginning is weak, and after 1 or 2 minutes he will be better and better and better. It is just reverse of it. It's the antibodies against pre-synaptic. It is the disease associated with the what we call paraneoplastic syndromes. That is the application, fine application. Another application, look, the application anarrhythmic drugs. Anti-arrhythmic drugs is the Vaughan-Williams classification. Class 1, 2, 3, 4, and not classified others. One is on sodium channel blockers or membrane stabilizers. For example, given only lignocaine, although they are definitely classified 1A, B, etc. They are again dependent upon the sodium channel. And 2, the beta blockers, they are also related with the channel, blocking the receptors. Receptor means that you are going inside, channels again are there. Third one is the potassium channel blockers, like amantadine. And एक चीज़ होती है पोटाशियम चैनल एक्टिवेटर्स, ये आपके लिए मैं छोड़ देता हूँ. एक चीज़ और भी होती है पोटाशियम चैनल एक्टिवेटर. वो क्या चीज़ होती है, वो ज़रा सा मुझे बताएगा कोई, आप में सुनने वाला हो तो मुझे जो है ना वो कर दे, इस पर व्हाट्सएप पे इसका जवाब दे दे. व्हाट आर पोटाशियम चैनल एक्टिवेटर वेयर दे आर यूज्ड. मेरा क्वेश्चन ये है कि वो इस्तेमाल कहाँ होते हैं, अगर हैं तो क्या है. ये तो आम दवा, कैल्शियम चैनल ब्लॉकर्स, CCP फेवरेट. तो जो पिरिमिडोन डेरिवेटिव या नॉन पिरिमिडोन डेरिवेटिव, वर्बामिल है, एंड इसी तरीके से एम्लोडिपिन वगैरह है. दीज आर क्लास 4. डिजॉक्सिन, एडेनोसिन, मैग्नीशियम सल्फेट, एट्रोपिन, इस पर वर्क किया जा रहा है कि इसके चैनल्स कौन से हैं, कि ऑल डिपेंडेंट अपॉन द चैनल्स. Specifically designed for digoxin, specifically designed for adenosine, magnesium sulfate, atropine. Specific, remember where I am using specific. The channelopathy, they will detect channelopathy, it means that it is very, very specific to this. But you can classify, you can apply this knowledge to the medicine now, pharmacology, agriculture, and others, you can apply. Now this is actually the part where I stopped previously, because the time I have taken much time has been taken by me, and that is why I will show you where I stopped previously here. Step 2, neurotransmitter causes muscle membrane gates to open. Now this is clear now. What are these gates? That is why I have talked so much about the channels are concerned. Step 3 is this: in the contraction is concerned, ions, sodium and potassium exchange place depending upon the channels, causing the sarcoplasmic reticulum to release calcium. Now, this is the resting membrane potential generation of it. Step 4, now the calcium will release again through its channel, starts the muscle contraction as the action. Contraction means that this is actually what is called action potential. By how it moves, the myosin filaments and actin filaments, they slide each other. Myosin has got 2 words. It grasps the actin just like a scissor, and then it slides to each other. This is why the myofibril contact. Now this is how the myofibril will contact. अब ये जो डिस्कशन है यहाँ तक किया गया है, अगर हम फर्स्ट करें आप किसी को बताएंगे तो कहेगा भाई इससे पढ़ने की क्या ज़रूरत थी. और पढ़ने की ज़रूरत इसलिए है कि मतलब आयंस के वर्ड आएगा आगे. अभी वर्ड में कि आप चैनलोपैथी इसके ऊपर सारा डिपेंडेंट है. ये अभी तक जो है वो चैनल्स पे डिपेंड करते हैं, वो उसी को डिस्क्रिप्शन लाजमी मांगते हैं. उसके बगैर जो फार्मास्यूटिकल कंपनीज को वो नई दवा जो बनाते हैं तो वो पूछते हैं कि चैनल्स कौन सा इस्तेमाल करते हैं. आप समझिए आ बात आपको, हाउ दे अप्लाइंग दिस. तो ये भी आपको पता तो होना चाहिए ना भाई ये क्या चीज़ होती है. इसी तरह से उन्होंने जो है ना कैल्शियम चैनल ब्लॉकर है, पोटेशियम एक्टिवेटर है. मैंने कहा आपसे एक्टिवेटर्स भी होते हैं इसमें और ब्लॉकर्स भी होते हैं. ब्लॉकर में तो एमाड्रोन हो गया था और एक्टिवेटर में रह गया था. वो आप ज़रा सा मतलब अपने जहन से सोचिए, किताब में ना देखें. अगर आप किताब में देखा तो निकल आएगा. आप सोचें, आपने जो पढ़ा हुआ है, ये पढ़ा हुआ होगा डेफिनेटली, तो इसको आप ज़रा से देखिएगा कि कहाँ पे इन्होंने नॉलेज ही अप्लाई की है, जो मैंने छोड़ के जा रहा था। और उसके बाद में अगला जो है ना सेशन यहाँ से उठाऊँगा, अब ये अपनी बात को फिर आगे करूँगा यहाँ पे। स्लाइड फिलामेंट थ्योरी ऑफ मसल कॉन्ट्रैक्शन, ये पूरा का पूरा इन्फ्लक्स ऑफ सोडियम कॉजेज द थिक मायोसिन फिलामेंट्स form cross bridges within this thing, actin filament by exposing the binding site on the actin. This is the last statement he has given. Here it is the see there, the ADP is used here. Myosin had high energy configuration with this. So actin is sliding on this. This is how sliding theory is there. And slide filament theory, the cross bridges change shape as it pulls the filaments which slides toward the center of the sarcomere in the power stroke. खाली ये नहीं हुआ कि उसने वो कर दिया क्रॉस होने से जो रिजल्ट बरामद हो रहा है वो ये हो रहा है कि वो शॉर्ट करेगा मायोफिब्रिल को क्रॉस ब्रिजेस से। तो डिस्टेंस बिटवीन द ये जेड लाइन जो कराती है ना, जो इसमें आप निकालते हैं इलेक्ट्रॉन माइक्रोस्कोप से, आप देखेंगे इस फिनोमेना को, जनरेशन को, तो आपको ये लाइंस की डिस्क्रिप्शन मिलेगी। वो जो लाइन है रिप्रेजेंट कर रही होती है, इफ द डिस्टेंस बिटवीन द जेड लाइन डिक्रीजेज इट मीन दैट मसल मायोफिब्रिल हैज़ कॉन्ट्रैक्टेड। उसके बिहाइंड ये सारी की सारी जो है ना ये नॉलेज आपको अप्लाई करनी पड़ेगी अगर इसको अच्छा तरह आना, अगर आप चाहते हैं। ये क्रॉस ब्रिज डिटैच फ्रॉम द एक्टिन फिलामेंट, उसके बाद वो डिटैच हो जाता है, काम खत्म हो जाता है। देन एटीपी बॉन्ड्स टू मायोसिन हेड, जब एटीपी बॉन्ड कर देता है मायोसिन के हेड को, तो क्रॉस ब्रिजिंग थी उसकी, उसका फायदा, उसका काम खत्म हो गया, दैट्स ऑल। वो रिलैक्सिंग में इसमें आ जाएगा। और इसी तरीके से मायोसिन हेड गेट्स रेडी टू बॉन्ड टू एक्टिन अगेन, फिर यूजिंग एटीपी एनर्जी, वो सेम साइकिल रिपीटेड एंड रिपीटेड एंड रिपीटेड। आप ये सार्कोमेयर पे अप्लाई करेंगे, देन उसके बाद बंडल पे अप्लाई करेंगे, उसके बाद फेसिकल पे अप्लाई करेंगे, फिर होल मसल पे अप्लाई करते हैं। तो कहाँ से बात ली थी, कहाँ पे ख़त्म हो जाती है, देखिए। अगर हम छोड़ देते हैं, फर्स्ट करें, ये इसी फिनोमेना को डिस्क्राइब किया जो मैंने अभी भी बताया कि किस तरह किसी इलेक्ट्रॉन माइक्रोस्कोप और पोलराइजेशन ऑफ लाइट से इन्होंने लाइन्स डिस्क्राइब की हैं। तो मसल कॉन्ट्रैक्शन की फिजियोलॉजी और मसल कॉन्ट्रैक्शन, जिसके मैंने डिस्क्रिप्शन दिया हुआ है, तो इसको हम यहाँ से ले लेंगे बाद में, और फिर इसको कंप्लीट करेंगे स्केलेटल मसल के लिए। थ्योरी यही रही है इसमें मसल में, उसके खाली न्यूरोट्रांसमीटर चेंज हो जाएंगे, और इसी तरह से जो कार्डियक मसल है उसके भी अंदर सेम थ्योरी है जो हम अप्लाई करेंगे ये अपनी नॉलेज को, तो वो भी जो न्यूरोट्रांसमीटर की वजह से मुख्तलिफ होती है, और उसकी मॉर्फोलॉजी के लिहाज से भी वो मुख्तलिफ हो जाती है, वो बाद में डिस्कशन में आएगा। इतना अगर आप पढ़ते हैं ना लेकिन डॉक्टर साहब, फिर आपकी मानता हूँ मैं कि नॉलेजेबल बनता है, भाई क्या बात है। लेकिन अगर आप झटपट निकाल देंगे, हमें क्या पता, चैनल पैनल हम नहीं जानते, और आगे बढ़ गए कंसल्टेंट बन गए, और आपको पता ही नहीं है ये है क्या चीज़। तो फिर वो जब कहीं गए आप, वहाँ उन्होंने ये बात निकाल दी, उन्होंने प्रोफेसर आदमी, एसोसिएट प्रोफेसर, उसको नहीं पता ये चैनलोपैथी क्या होती है और कहाँ से वजूद में आई है। तो ये मैं कोशिश करता हूँ कि जो भी इसमें इंटरेस्टेड हैं ज़रूर इसको देखें प्लीज़। ये आपके काम की चीज़ है, स्पेसिफिक लोगों के लिए, नॉट ऑल। जो इसको इंटरेस्टेड लोग ज़रूर पढ़ें। फिर इसका सेशन 3 में इसको कंप्लीट करेंगे और फिर आगे बढ़ेंगे। हमारा इस तरह होता है पढ़ना, जिस तरह हमने पढ़ाया है उसी तरह आपको बताया है, ठीक है जी। तो वमा अलैना इल्लल बलाग, इंशाअल्लाह सी यू सून इंशाअल्लाह ताला। अस्सलामु अलैकुम व रहमतुल्लाहि व बरकातहू।